This video will cover question 62 from the biology section of the 2018 NSAA paper. In this question, we're told that an epithelial cell from an animal was removed and that the cytoplasm of this cell could be considered as a 2% sugar solution. The cell was then placed in 4% sugar solution, so this is a more concentrated sugar solution than the cytoplasm, and then we're asked which of these statements is correct. So before we get into the statements, we need to think about what's going to be happening when we have a situation where we have our animal cell with a lower glucose concentration than the surrounding 4% sugar solution. A way to think about it is to think about the different fluid-filled regions consisting of water molecules and sugar molecules. Now, the animal cell cytoplasm is more dilute. That means the water molecules are more free to move than they would be in the more concentrated sugar solution. And that brings us to what will happen next. Water will move out of the cell via osmosis, down a water potential gradient. And water potential means how free water molecules are to move. So naturally, in the more concentrated solution, the water molecule's movement is more impeded by these solutes, whereas in the more dilute cell, there's a higher water potential. So there's movement down that potential across a semi-permeable membrane. Also, it's worth noting that since there's a higher concentration of sugar on the outside than on the inside, there might be some diffusion of the sugar into the cell. Okay, so that's what we can glean would happen from the information. Let's look at our statements. At equilibrium, the sugar concentration in the cell was 6%. So at equilibrium, the point at which this movement down via osmosis or perhaps via diffusion, we're told that equilibrium point would be when the sugar concentration is 6%. Well, we can know that's wrong because the cytoplasmic sugar concentration is 2% and the surrounding solution is only 4%. So there's no way in which you can get to 6% within the cytoplasm without there being a more concentrated solution of sugar introduced into the experiment. So there's no way you can get to 6%. Moving on to 2, water continued to move across the cell membrane after equilibrium was reached. Well, we know this to be true because osmosis is the net movement of water molecules down a water potential gradient. Water molecules are moving to and fro on either side of a membrane all the time. The difference is that when there's a difference in water potential, overall, the traffic is generally down the water potential gradient rather than against it. So even after equilibrium, there will be interchange of water molecules, but there will be no change in the water potential on either side. Moving on to statement 3, osmosis was most rapid when the cell was first placed in the solution. Well, if we think about it, when the cell was first placed in the solution, then it will have had that 2% concentration versus the 4% surrounding it. Over time, water molecules would leave, thereby reducing the water potential within the cell and therefore making the gradient less steep. So yes, the gradient of the difference between water potentials would have been at its greatest when the cell was first introduced into the solution. So we have our answer that 2 and 3 are correct statements, and that gives us our answer as option G.